and welcome everybody to my channel Umini Bio Entrance, and today we will be discussing CSIR Net JRF Life Science question paper. This is shift 2 question paper and I am discussing the part C section, and in this video I will be discussing the biochemistry questions which they have asked in shift 2 part C section. This was conducted in the year March 2025. If you are new to my channel, please do like, share and subscribe. If you want still previous year question paper of CSIR Net Life Science being discussed, you can go to my channel Umini Bio Entrance. In the channel playlist, select CSIR Net exams and there you will find. So coming to the first question. In a typical experiment, 15 ml of an aqueous solution containing an unknown quantity of acetylcholine had a pH of 7.65. Okay. When the solution is incubated with acetylcholine esterase. Okay. So acetylcholine is there already in the aqueous solution. Then you add acetylcholine esterase and the pH of the solution decreased to 6.87. Assuming that there was no buffer in the reaction mixture, determine the number of moles of acetylcholine in 15 ml solution. So that is the question that they have asked. So you have to determine the number of moles of acetylcholine in the 15 ml solution. So here first what they have done is acetylcholine was taken, to that acetylcholine esterase was added and the pH has decreased. Why? Because acetylcholine will be reacted by acetylcholine esterase to form choline and acetic acid. Okay. Now this acetic acid is the one which is causing, or the formation of acetic acid is the one which is causing a decrease in the pH of the solution. Okay, now what they have asked, determine the number of moles of acetylcholine in the 15 ml solution. How many moles of acetylcholine is there? Since 1 mole of acetylcholine produces 1 mole of acetic acid, and we assume that this acetic acid, it will dissociate to produce 1 mole of H plus ions. Okay, so the number of moles of acetylcholine will be equal to the moles of H plus ions produced by the acetic acid. Got it? So that is how you have to calculate. Now what they have given? They have given only the 15 ml solution and 2 pH values they have given. So we know that pH is equal to minus log concentration of H plus and H plus is equal to 10 raised to minus pH. Now initial pH value and final pH value they have mentioned. So the decrease in pH is due to the production of acetic acid. I already told that. So from initial hydrogen ion concentration, how much it was? 2.24 into 10 raised to minus 8 molar. And final it is 1.35 into 10 raised to minus 7 molar. Then you have to see for the change in the hydrogen ion concentration. So final hydrogen ion concentration minus initial hydrogen ion concentration will give you the change in the hydrogen ion concentration. So the change will be 1.126 into 10 raised to minus 7 molar. Now I told you 1 mole of acetylcholine will be equal to the moles of H plus produced. So what will be the mole of H plus produced? They have given the concentration in molarity. So that will be equal to concentration into volume, or molarity into volume. So it will be 1.126 into 10 raised to minus 7 molar into they have given the volume as 15 ml. You have to convert it into liters because molarity is for liters, not for ml. So 0.015 liters. So that will give you around 1.65 into 10 raised to minus 9 mole into 1.75 into 10 raised to minus 9 mole. And that will be your option 1 as the answer. Okay. Now next one. Match the following ribose sugar pucker in nucleic acids labeled A, B, C, D with their corresponding conformational states. The black circle denotes the base of the nucleotide. So this is the base. This is the sugar puckering conformation they have given. Now from these you have to see whether A is C2XO or C2 endo, or whether it is O4XO or O4 endo or whatever. Now what is all this C2XO endo, O4, what is all that? So first coming to the explanation of all these things. So sugar puckering, it is the 3D conformation of the deoxyribose sugar, and in the DNA you cannot 100% tell that the sugar is always in a flat planar surface. No, it will be having its carbon atom arranged out of the plane of the paper. Okay, it is not flat. It is a three-dimensional conformation. Okay, that is called as sugar puckering. Now, why does this DNA need this sugar puckering? It is for spacing of the between the base pairs. Then different sugar pucker conformations are there in different forms of DNA, and because of that there will be difference in the width and depth of the major and the minor groove. Then sugar puckering also will help in the flexibility of the DNA, and it also help in the interaction of proteins and enzymes that are needed for DNA replication or transcription, etc. Now what is C3 endo conformation? Now C3 endo conformation means, it is very simple to understand that the 3' means the position of the carbon atom, C is the carbon atom, 3', that means the carbon atom at position 3. That is what is meant by C3', because in a DNA molecule the numbering for the sugar is 1', 2', 3', 4', 5'. Okay. So it is C3' means carbon atom at position number 3 is in endo form, endo conformation. That means it is raised above the plane of this paper. See here, 3' endo. It is raised, and above that the 3' endo conformation, it is lying in the same side as your base. See here, it is raised above. It is on the same side as your base. That is C3' endo conformation. Now if it is C2', then it is the carbon atom at position 2, that is raised above the plane of the paper. So it is 2' endo, and the 2' endo is lying on the same side as your base and is near to the base. So it is C2' endo conformation. Fine. That is with regard to the C3' endo and C2' endo. Now what is exo conformation? See here, exo conformation means the carbon atom that is displaced is on the opposite side of the base. Okay. Now this is C2'. This is the second carbon atom. Okay, this one. This is C2' exo conformation. That is, the second carbon atom is below the plane and away from the base. See, this is the normal planar flat form of your deoxyribose sugar. This is the base. This is the 3' carbon. This is 5'. And this one is your C2 carbon. Now C2 carbon is below and away from the base. It is below the projected below the plane of the paper. So it is below this and away from the base. So it is exo conformation. Okay. This one C3', this is above and on the same side as that of your base. That is your endo conformation. Now this is C3' exo conformation, below the plane of the paper and and away from the base, opposite side of the base. So it is C3 prime exo conformation. Now coming to what is called as O4 prime exo and O4 prime endo. Now O means in the sugar you have only the O over here, that is oxygen molecule. That is this. Now so O prime exo endo, it will be based on the position of the oxygen atom in the sugar molecule. Now if the oxygen atom is attached to the sugar is positioned above the plane, that is like this, okay, above the plane, and it is on the same side as your C5 carbon atom, then it is endo conformation. So oxygen atom in relation to C5 carbon atom, it is placed above the plane of the sugar, then it is an endo conformation. Now if it is below and opposite the C5 carbon atom, then it is exo conformation. Okay, so that is what is meant by this sugar puckering. Now we will see the question. Now coming to the question. Match the following ribose sugar pucker. Okay, this is not a deoxyribose molecule. They have given a ribose sugar. Okay, now it is in RNA. Ribose sugar pucker in nucleic acids and their corresponding conformational states. The black circle denotes the base of the nucleotide. So here is your black circle, here is your C5 carbon atom. Now you have to match what each these sugar pucker denotes. Now they have told A2 is C2 prime exo. So carbon, yes, 2 is here. It is not exo, it is endo. It is on above the plane of the sugar molecule, and it is lying on the same side as your base, and it is very near to the base. So it is C2 prime endo. So that makes option 1 gone as well as option 4 gone. Now we have to see between 2 and 3. Now B, they have told O4 prime endo and O4 prime exo. Okay, so we have to see. O means this one. Now the position of O is what matters. So O is here. So O is on the same side as that of your C5 atom. See here, C5 atom is here. O is also projected almost the same side as your C5 atom. So it must be endo and not exo conformation. So that makes option 2 as your answer, and option 3 is not correct. But I will tell you the others also. Okay. Now C is C3 prime endo. Endo means above and on the same side as that of your base. So 3 is above and on the same side as that of your base. So that is C3 prime endo. Now what will be D? D is okay based on the position of the this thing. What is that? Oxygen. Because it is O4 in all the options. So O4 and the oxygen atom is below the plane and it is away from or lying opposite side of the C5 atom. So it has to be exo conformation. So D is O4 prime exo. So answer is option 2. Then given below are 4 metabolic intermediates listed against amino acids A to E. So these are the amino acids, these are the intermediates. Now which one of the following option correctly pairs the metabolic intermediates with their corresponding amino acid end products? Now these are certain intermediates that are involved in the biosynthesis of these amino acids. Now which intermediate will give rise to which type of amino acid, you have to match from these. Okay. Now see here, You have the intermediate like 3-phosphoglycerate, pyruvate, PEP plus erythrose 4-phosphate, alpha-ketoglutarate, oxaloacetate, ribose 5-phosphate. These are the various intermediates that are involved in citric acid cycle as well as your pentose phosphate. So these intermediates, they are involved in the synthesis of various amino acids. So 3-phosphoglycerate, it is involved in serine, glycine, cysteine, and it is a 1, 2, 3, 3 carbon compound. Okay. Then you have pyruvate, again a 3 carbon compound, but it is having C double bond O here in the center. So that is your pyruvate, and it is involved in aline, valine, leucine, isoleucine synthesis. Then you have PEP, that is your phosphoenol pyruvate and erythrose 4-phosphate. Erythrose 4-phosphate means 4 carbon compound. Phosphoenol pyruvate, again a 3 carbon compound, but it has a phosphate group attached over here. So that is involved in your aromatic amino acids, that is phenylalanine, tyrosine and tryptophan. Then you have alpha ketoglutarate 1, 2, 3, 4, 5 carbon compound, and it is glutamic acid, glutamine, proline and arginine. Then you have oxaloacetate, a 4 carbon compound, and this is different from erythrose 4-phosphate, where a phosphate group is attached to the 4th carbon atom in erythrose 4-phosphate, but it is not there in oxaloacetate. And moreover, oxaloacetate have got 2 carboxyls at their ends. And these oxaloacetate, it is involved in aspartic acid, asparagine, methionine, threonine, lysine. And ribose 5-phosphate, a phosphate group attached to 5th carbon atom, it is involved in the synthesis of histidine. Now these things you have to be very clear with regards to the structure of the various intermediates as well as what all amino acids they are involved in the synthesis. Okay, this you have to clearly learn. Only then you will be able to answer that question. Now see this one. 1, 2, 3, 4, 5 carbon compound, 3 carboxyl groups. We have not encountered that with the 3 carboxyl groups and 5 carbon compound. So it is not involved in the synthesis of any amino acids, and this structure, it is that of citrate. Okay. So citrate is not involved in the synthesis of any amino acid. So that makes option A gone. So wherever A is coming, that is not the correct answer. Okay, then you have 4. Okay, 4th, this one. Yeah, this one, a 3 carbon compound, and it is pyruvate, C double bond O, CO, CH3. It is pyruvate, and pyruvate, I told you, it is involved in alanine, valine, leucine synthesis. So B is correct. That means 4 also gone. So your answer is option 2, B and D. Okay, I will tell you the others also. Then third one, this one. This is they are telling it is involved in glutamate, glutamine, proline. But actually this is not the one which is involved. We have not encountered SCOA molecule attached to a carbon skeleton. Okay, which is involved in the biosynthesis of amino acids. So this molecule must be glutaryl CoA. And if it is glutaryl CoA, then it is involved in the breakdown of amino acids like lysine, hydroxy, lysine and tryptophan. So it is not involved in any biosynthesis activity. So that makes option 3 also wrong. Then the second one, that is they are telling it is involved in the synthesis of methionine, threonine and lysine. So that must be this one. Okay, 2 carboxylic groups. Okay, then the C double bond O is there. So it is an oxaloacetate. and it is involved in the synthesis of aspartic acid, asparagine, methionine, threonine, lysine. So that makes D correct. Then E1, okay, 1 histidine. This is not, I told you 1 is citrate, and histidine, it is ribose 5-phosphate that is involved. So that makes E also wrong. So your answer is option 2, that is B and D, which are the right answers. Then next one, the graph below shows the plot of 1 by V versus 1 by S, that is your Lineweaver-Burk plot, for an enzymatic reaction with the solid and dashed lines representing reactions without and with an inhibitor. So this line is without inhibitor, and this line is with inhibitor. The concentration of the inhibitor is 1 micromolar that they have given. Now, which of the following will be the Ki of the inhibitor? Now, if you see the graph, you can see that there is no change. This is your Vmax and these are your Km values. Okay. This is your Vmax and these are your Km values. So here there is no change in the Vmax. It is unchanged. But Km value, if you see, without inhibitor it is -3 and with the inhibitor it is -1. That is, Km value has increased. So it is the case of a typical competitive inhibitor. Okay. So in competitive inhibitor, what is happening is the inhibitor molecule will compete with the substrate for binding to the active site of the enzyme. So that is competitive inhibitor. Now what they have asked, we have to find the Ki of the inhibitor. So we know the formula, that is alpha is equal to 1 plus concentration of I by Ki. Okay. So alpha minus, take this one this side of the equal sign, then alpha minus 1 is equal to minus I by Ki, or Ki is equal to concentration of I by alpha minus 1. Now what is this alpha? Alpha is Km dash by Km. Now this value, it is 1 by Km. Okay. These are 1 by Km values from the graph. They are not just -3 and -1. They are 1 by Km. So for the inhibitor it is Km dash, and for the without inhibitor it is your Km. So here it will be -1 by -1 and here it will be -1 by 3. So that will give you the value of alpha, that is -3 by 1, that is 3. Then Ki is concentration of I by alpha minus 1. So that will be 0.5 micromolar. So your answer here is option 3. Now before coming to the next question, I will tell you what is a CD spectra. So CD spectra, it is for providing information regarding the secondary structure of proteins. And secondary structure, you know, there are alpha coils and helices, beta turns and beta sheets. Now regarding whether it is in alpha helix or beta, or whether it is a random coil, you will get information from the CD spectra. And this is the classical or the standard secondary structure peaks that you get for in the CD spectra for secondary structure of proteins. Now if there is any deviation from this classical structure peak, then you can say it could be a mixture of alpha and beta. Now if you see this graph, you can say that it is starting from around 190 or 160 to and is extending till 250. So this wavelength region is where you will get the secondary structure graphs, that is alpha, beta and of random coil. Okay. Now what is this alpha helix? You can see that it is having a positive peak over here at a wavelength range of around 190 nanometers, C alpha. And there are two negative peaks. One is this peak and the other is this peak. So this first peak is around nearly around 208 nanometers, and the second peak will be somewhere around 222 nanometers. That is always maintained at that range in case of alpha peak. Now if you take the condition of beta peak, it is having one positive peak and one negative sheet. So the negative peak, it will be somewhere in the range of 216 to 218 nanometer range, and the positive peak, it will be somewhere around 195 to 198, like that. Now if it is in the random coil conformation, then you have one negative peak and one positive peak, and this negative peak, it will be around 195 nanometer range, and the positive peak, it will be around 200 nanometer to 280 nanometer range. That is in the case of random coil. Now see the question. The circular dichorism spectra for near UV and far UV regions of a polypeptide chain are given below. So this is with the case of your secondary structure of proteins. Okay, that is, I told you it is in the range from 190 to 250, like that. So here also it is coming from that range to 250. Okay, so this is the first graph showing the secondary structure of the protein. But this graph, if you see, it is starting from 250 and going till 330 nanometer range. So that will be your tertiary structure. Okay, this is the CD spectroscopy for the tertiary structure, and this is the near UV range, 250 to 330 nanometer. That is the near UV range. And this region, they tell it is that is a tertiary structure. It is sensitive to aromatic amino acids like phenylalanine, tyrosine, tryptophan, as well as to disulfide bonds. And you know, these disulfide bonds, they are involved in the tertiary structure of the protein. And if there is any change in this graph, that is if there is a change in the intensity or the position of these amino acids from their characteristic CD spectra, then you could suggest that there is a change in the tertiary structure of the protein. Okay. Now you can get information by comparing it with the secondary structure graph, and you can tell whether it is an alpha plus beta or alpha beta structure. Okay. Now here if you see which one of the following option represent a correct inference about the polypeptide fold based on the above data. Okay. It contains only beta sheet. Now if you see this graph, it doesn't show a proper alpha helix. See here, this is the helix CD spectra for an alpha helix, and that is for a beta helix. Now if you see here, it doesn't show a typical alpha helix. It's not like what you see here, nor is it a beta helix, a beta sheet. So you cannot confirm and tell 100% it contains only beta sheets. No, it can be a mixture of alpha and beta. I told you already, if it is deviating from the classical secondary structure, then it can be a mixture of alpha and beta peaks. So that makes the first option wrong. Then what is the next thing they have told? It has to be an alternate alpha beta fold. How can you confirm and tell by without giving much information about the peak in the tertiary structure, and neither in the secondary structure? You cannot 100% tell that it has to be. You are telling it with full confidence. That is what is meant by it has to be alternate alpha beta fold. You cannot tell that. Then third also, again, it has to be. Okay, that again makes it wrong. Now you can tell, okay, it belongs to either alternate alpha beta or mixed alpha plus beta fold. You cannot tell by just seeing these two graphs without any much information regarding the peaks. Okay, so 4 will be the answer. In general, you cannot conclude anything just based on these two graphs. More information is required. Now, delta G0 of a reaction shows the following temperature dependence. Now, if you see the graph here, you can see that the delta G0, it is a constant value, okay. Now, delta G0, it is not changing with the temperature. Here, the temperature increases, delta G0 is remaining constant, no change at all. Okay, now what is what they have asked in the question? What is the expected dependence of K equilibrium of the reaction on the temperature, where C is the temperature independent constant? Okay. Now see the calculation. Now you know that there is a relationship between delta G0 dash 0 and K equilibrium, and it is given by minus RT ln K equilibrium. We know from this graph delta G0 is a constant. So constant is equal to minus RT ln K equilibrium, or ln K equilibrium is equal to minus constant by RT, or I can split it that R is also a constant. Only the temperature is changing there. So minus constant by R into 1 by T, and this thing minus constant by R, I am taking it as a constant, into 1 by T. Now you have to change this ln, and you can use the E value here. So K equilibrium is equal to C into E raised to 1 by 2. So that makes the answer 2 as your answer. So that's it with regards to the biochemistry questions, what they have asked in part C. Thank you for watching. Please do like, share and subscribe if you are new to my channel. If you want still previous year question papers of biochemistry being discussed, you can go to my channel Omni Bio Entrance. In the channel playlist, C for CSIR net exams, and there you will find. Thank you.